So in Chapter 9 we talked about the structure of DNA, and if you remember when Watson and Crick discovered the structure, it was immediately apparent to them just how DNA would replicate. And so Chapter 9, or Chapter 10 rather, is about exactly how DNA replicates. So here you have a picture of a bunch of identical twins. In a human body, or at least in an adult, we have about 65 trillion cells. In most of our tissues and most of our cells in those tissues, we have nuclei, and within those nuclei we have our DNA. If these individuals are identical twins, right, they came from, they should have, right, more or less the same DNA. In order to produce all 65 trillion cells, or close to that 65 trillion cells, with exactly the same DNA, how much fidelity should there be in the system? Like, how good could it be without a mistake? I mean, it seems pretty out there, or rather pretty challenging to have that happen. And in fact, the answer is that it doesn't. So they probably share the same genes, but possibly not the same version of each of those genes. This chapter is pretty straightforward. We're basically just going to look at DNA replication in vivo and in vitro. So three basic features of DNA replication is that it's semi-conservative, it has unique origins where it initiates, and it proceeds in a bidirectional fashion. We'll talk about these right now. So semi-conservative means that each of the parental strands is conserved and serves as the template for the synthesis of the new complementary strand. Watson and Crick first proposed this mechanism of DNA replication. All you need to do is unzip the DNA molecule by breaking those hydrogen bonds that are formed between the complementary base pairs, exposing the parental templates to a whole bunch of nucleotides and some enzymes, and it's a pretty straightforward process, and you're off and running. There are, however, other potential modes of DNA replication. For example, here, conservative DNA replication, you would start with the parental DNA. The first generation progeny, however, as opposed to the semi-conservative DNA replication where you have one of the templates that originated in the parental DNA for each of the two progeny, with conservative DNA replication, you have one of the progeny that has both of the parental templates and the other progeny that has both of the new templates. The next generation, as you can see there at the bottom, you would have one progeny in the second generation that is essentially identical to the parental DNA strands, and then three that actually are composed of newly synthesized DNA strands. Another type is dispersive DNA replication, where, as you can see in the first and second generation progeny, you just get a smattering on each of the templates of parental DNA and then also newly synthesized DNA. And so there are a bunch of potential modes of DNA replication. Watson and Crick believed semi-conservative replication was, seemed to be the most straightforward way, but we don't really know, right? How at that point we have potential ones, one that seems theoretically most straightforward, but what we really need to do is have experimental evidence that supports, one of these or the other. And so try to think of a way that or some sort of experiment that you could perform there where you could distinguish between these different modes of DNA replication. So in 1958, Matthew Meselson and Franklin-Stahl showed that DNA replication in E. coli is semi-conservative, And this is a figure that just shows the general idea of their experiment. So the technique they used was something called density gradient centrifugation. If you look there on the right, those are basically picture those things in the purple as centrifuge tubes. When you centrifuge something of something, then the heavier things will fall out closer to the end because they're heavier. And so basically, what they wanted to do is they wanted to see if they could separate out light strands of DNA from heavy strands of DNA. And so, in order to produce those heavy strands of DNA, they grew E. coli in a medium that contained nitrogen 15. So, the normal atomic number for nitrogen is 14, so nitrogen 15 is heavier than the normal nitrogen. When a cell is reproducing, nucleotides, the nitrogen-containing bases on the nucleotides will contain nitrogen. And so if you're growing them in nitrogen 15, the assumption is that the DNA strands, the new DNA strands that are being grown, will be grown with bases that contain the heavy nitrogen. Therefore, those strands should be heavier. So after you grow those cells with the heavy nitrogen, you extract them, you put them in the centrifuge, and there on the upper right, you see the result. If you look at the yellow band there, that is the newly formed, but since it was a bunch of generations, we're calling it the parental DNA, right? The parental DNA then is a heavy form. In order to contrast that with a light form or the natural normal form, you see the band that is just to the left of the yellow one on the control, that third one down, and that's just your normal sort of DNA. And so now you have a heavy strand of DNA and a light strand of DNA. Okay, so that was step one of the experiment. Step two then would be to transfer the cells to a medium that has nitrogen 14, the sort of normal nitrogen, the light form of nitrogen, right, the natural light form of nitrogen, and grow them up for one generation. As they grow up for one generation, if your DNA replication is semi-conservative, as you see there on step 3, you then should produce a sort of hybrid strand of DNA where one of the strands is a heavy strand and one of the strands is a light strand. And so then when you go through, you extract the DNA and you spin that DNA, because one of the strands is light and one of the strands is heavy, you should get a band that is somewhere in between the heavy and light strand when you put them through the centrifuge. And that's exactly what they found after one generation. Then for the second generation, if you look there on step 5, you now have each of the hybrid strands, right, producing, separating out, serving as templates. But then one of the hybrid strands, right, is the light form. It serves as a template. You again are still in a medium that has the light nitrogen. So what that results in is two, if you look there under step 5, is two DNA molecules that have both light strands and two DNA molecules that have one heavy strand and one light strand. You go through and you spin that one, and you get a light band and a hybrid band. And then if you keep going for more and more generations, you should get accumulation of light bands and a maintenance of just the same sort of hybrid band. So it's a really sort of straightforward, pretty easy experiment to do. It's one of those sort of elegant, simple, straightforward experiments that you see that are often very, very influential. So here's a simplified version of DNA replication. Essentially, all you need to do is unzip those strands, expose the bases to nucleotides, throw in a bunch of nucleotides, throw in a bunch of enzymes to catalyze these reactions, and once those new nucleotides here in light blue find and pair with their complementary base pairs. You then use the enzymes to sort of put it all back together, and you have a new strand of DNA that has been replicated in that semi-conservative form. But where does it start? That's sort of our next question. Potentially, we could have replication starting at one place. We could have replication starting at two places, at three places, at multiple places. And so researchers in the 1960s were addressing this question. One researcher, John Cairns, grew E. coli in the presence of heavy hydrogen, thymidine, one of the base pairs, right? And he did that so that it's radioactive, and he did that so that he could then expose it to audio radiographs, and he could see the new DNA strands grow in sort of real time by varying the periods of time that he lets them grow. So he lets them grow for a certain amount of time, then a little bit longer, a little bit longer, a little bit longer. At each of those, in a way, he can take sort of snapshots of the DNA growing. And so this was a way to visualize the replication of the DNA. And as you can see here in the diagram, it clearly sort of shows that DNA replication occurred or began at a single place. We later discovered that it wasn't unidirectional, but rather it was bidirectional, and that sort of made sense because we knew that DNA was replicating fast, and replicating in a bidirectional fashion is much faster than a unidirectional fashion. Okay, so now the question becomes, what are the characteristics of that area where DNA replication begins? When we begin to look at lots of these areas, what we found is that they had certain things in common. What we found is that they had these tandem 13 base pair long sequences that shared characteristics. So if you look there in the blue, those are those 13 base pairs that these sort of tandem repeat areas or regions had in common. You had GACT-TN. Here, N just means nucleotide, so that could be any of the four bases there, and so that position, what we call the position, could vary, but then it would be followed by two T's and then another position that could vary, and then four more T's. We also found that there were four sites where the initiation proteins would bind. When we looked at the base pair composition of those four sites, there you see in the red, it would be TTA-T, again N, it could vary there, and then a CA, again another site that varies, and then an A. And these were all 9 base pairs long. So what we notice about both of these regions is that they are rich in A's and T's. And so then the question becomes, okay, how come they're rich? What does that tell us? And if we think about the hydrogen bonds that form between G's and C's, or A's and T's, we should remember that when G bonds with C, there are 3 hydrogen bonds, and when A bonds with T, there are only 2 hydrogen bonds. So this makes sense to us. With only 2 hydrogen bonds, the 2 strands are going to separate out in an easier fashion, and so you can begin replicating in those areas. So in E. coli, when we see DNA replicating, we call this a replication bubble. And when we begin to look at other bacteria and viral chromosomes, which are small, we would usually see it a single unique origin of replication. But they're small. In eukaryotes, because they're much larger, and because they're separated in multiple chromosomes, what we saw is that there was not a single origin of replication, but rather multiple origins of replication. In humans, just for example, there's about 10,000. And this makes sense. If you have that much DNA to replicate, if there was just a single site of origination, it would take I mean, at a minimum, you would have to have obviously a single site at each chromosome, but even within those chromosomes, it would just take way, way too long. In eukaryotes, we call that sort of that sort of unit where the DNA becomes replicated on the chromosome. We call that, just for terminology's sake, a replicon. So here are the key points of this section. So in this section, we're going to look at DNA synthesis in vitro. A lot of what we know about DNA synthesis is stuff that we've deduced from in vitro studies. We're going to begin by talking about DNA polymerases. So ACE, whenever you see polymerase, that means it's an enzyme. Polymer basically means it's forming a polymer. In this case, the polymer that is forming is DNA. And so enzymes that form DNA. The first DNA polymerase was discovered in E. coli by a guy by the name of Arthur Kornberg in 1957. He was actually able to isolate this enzyme and expose that enzyme to nucleotides, and that enzyme would then synthesize DNA under certain circumstances. Originally, this enzyme, bore his name, and it was called Kornberg's enzyme. What we found is that there were certain requirements for DNA polymerase. One important requirement is that the polymerases needed to have access to a hydroxyl that came off of the 3' carbon on a primer strand of DNA. And we'll talk about what a primer strand is here in a second. It also needed a template, obviously, to specify which nucleotide needed to be inserted next. And then also then the ingredients. The substrates, we're calling them DNTPs. These are deoxy. N is the nucleotide. At the bottom there you see thymidine, a thymidine example, triphosphate. So that's basically your nucleotide. In that triphosphate, you have the energy to form the bond between the last nucleotide that has been added and that 3' hydroxyl, and the new nucleotide coming in, and that's basically just what this figure shows. Importantly, you need to remember that the growing strand of DNA goes in a very specific orientation. It grows from the 5' end to the 3' end. Okay, here's a diagram that shows that deoxythymidine triphosphate there at the bottom. You see the nitrogen-containing base there in green. You see the sugar there, sort of a tan color, and then you see the triphosphate there on the left. The phosphodiester bond forms when that phosphate attaches to that hydroxyl there, the 3' carbon of the sugar. So you see the guanine and the cytosine all the way at the top. There's that 5' end, so that you see that 5' carbon. I think we talked about this before, but just as a reminder, in that sugar, if you look at the different carbon atoms in the sugar, you see there's a 1', 2', 3', 4', and then a 5'. And so that's where this 5' end of the DNA strand and the 3' end of the DNA strand is coming from. And so at the 3' carbon, you have a hydroxyl that's attached to it. That hydroxyl is going to interact with that phosphate, and you're going to form that phosphodiester bond. And essentially, you are going to add a single nucleotide to that growing DNA strand. Again, very important, the DNA polymerase activity is in a 5' to 3' direction. So that means that it's reading the template DNA in the opposite direction, in a 3' to 5' direction. And so you need to to remember and understand that. It was also found that DNA polymerase 1 had an exonuclease activity. So exonuclease activity is basically the reverse of polymerase activity, where it will lop off or cut off a nucleotide or nucleic acids at the end of a strand. So here this diagram shows a 5' to 3' exonuclease activity there at the top. This figure shows that DNA polymerase 1 also has a 3' to 5' exonuclease activity that could be used to correct for base pairs that had been inserted incorrectly. Soon, though, even though this was the first polymerase that was characterized, it soon became known, or people figured out, that it probably wasn't the polymerase that was responsible for replication in E. coli, because they knew that DNA replicated in E. coli at a rate of about 1,000 base pairs per second, but DNA polymerase is only able to add 10 to 20 base pairs per second. And so the search continued for a polymerase that was responsible for DNA replication in E. coli, and that DNA polymerase ended up being DNA polymerase 3. But first, before we talk about the specifics of DNA polymerase 3, let's just look at all of the different polymerases. Well, some of all of the different polymerases that are out there. In E. coli, these are the ones that we're most familiar with. We have DNA polymerase 1, 2, 3, 4, and 5. For DNA replication, the main one for DNA replication is DNA polymerase 3. DNA polymerase 1 performs DNA replication when it is filling in between Okazaki fragments, where the primer gets replaced with DNA. For DNA repair, we have DNA polymerase 2, 4, and 5. Later on, we'll go through the whole process of DNA replication and see. I'm not going to, overburden you sort of with knowing all of those different ones, but just a couple of, main ones when we go through that process of DNA replication in E. coli. In eukaryotes, there's a lot more. It's more complicated. It's more complex. When you're replicating nuclear DNA, we find that DNA polymerase alpha, beta, and epsilon is involved. In replication of the mitochondrial DNA, that is the function of polymerase gamma. And then for DNA repair, we have polymerases beta, epsilon, kappa, zeta, mu, and nu. So for the exam, you want to make sure you know all of those. Just kidding. Okay, don't worry about the polymerases in the eukaryotes. But I just wanted to sort of throw them out there just to show you that it's a lot more complicated and complex out in the real world. All of these enzymes synthesize DNA from in a 5' to 3' direction again, and require a free 3' hydroxyl to start that to start the growing of that DNA strand. That comes from the primer. The primer is this tiny section of double-stranded DNA that is required to begin forming that complementary strand. This slide shows you a little bit more detail of DNA polymerase 3. It's made up of lots of subunits here. 2 beta subunits form this kind of sliding ring that's sort of in the advanced part of the DNA polymerase. We will show you DNA polymerase 3 in action in a bit, but one thing we should point out is that it is able to replicate at a very, very high fidelity. By fidelity, we mean how many times does it make an error? And what we see is there's usually only about one error per every billion base pairs, and so that's pretty good. Part of that is because we have proofreading mechanisms. Here, this diagram just kind of shows you what happens. In the proofreading, you need the exonuclease activity followed by the polymerase activity. And so here you have a primer terminal mismatch there, all the way on the left, where you see an A has been inserted, where a G should be inserted, and so then the polymerase comes along, DNA polymerase 3, sees that, takes it out, takes out the A, and then inserts the G, and then moves on. The DNA replication happens at different speeds. In humans, it's about 3,000 base pairs per minute. In bacteria, it's about 30,000. It can be 30,000 base pairs per minute. One other enzyme that we've talked about before from TAC, from that thermophile bacteria, and one that we'll talk about more, maybe we haven't talked about it yet, but one that we will talk about more when we talk about the PCR reaction, is an enzyme called TAC. Its error rate is actually not, doesn't have such high fidelity. It has an error rate of 1 base pair mismatch per every 9,000 base pairs. Okay, so here are the summary points about, with regards to the polymerases. One last one. Okay, you ready? Let's talk about the actual process of DNA replication. It's complex and it requires the concerted action of a whole bunch of different proteins. When all of those proteins come together, we call it the complex replication apparatus. So when DNA replication begins, you have replication occurring on both strands. One strand is called the leading strand and one strand is called the lagging strand. On the leading strand, the DNA synthesis is continuous. On the lagging strand, it's discontinuous. On this discontinuous strand, or the strand where discontinuous replication is happening, new DNA is synthesized in short segments. These segments are called Okazaki fragments. Later on, those segments are joined. In a second, we'll sort of demonstrate why it's necessary to just synthesize this new DNA in just these short sort of segments rather than continuously like we see on the leading strand. But for now, just remember, there's two different strands. The DNA strands open. There's one of the strands becomes the leading strand. The other strand becomes the lagging strand. Here are the essential or the important enzymes that are required for DNA replication. I've listed them with their primary function. So I want you to know and be familiar with each of these. Helicase, primase, single-stranded binding proteins, what the functions are. DNA urease or gyrase. I think probably the correct pronunciation is urease, like a euro. It's a Greek word. DNA polymerase and then DNA ligase. So here are the basic steps of DNA replication. Enzymes come in and open up the DNA molecule. First, you have to unwind the duplex, right? This is accomplished through helicase. Once it kind of becomes unzipped, you want to stabilize it so that it doesn't, instantly rezip. You do that with the single-stranded binding proteins. Because it's a double helix, as you are going through and sort of opening up, you're going to produce torque. DNA urease is the enzyme that sort of relieves that torque. Okay, once you have your your DNA molecule unzipped. You have the two different strands. In order to begin replicating, one of the requirements of those polymerase enzymes is that it starts from a double-stranded area. And so to have a double-stranded place to start that has that exposed 3' hydroxyl, you need to construct a primer, and that's done through RNA primase. So RNA primase comes in and constructs that primer. Then you have DNA polymerase 3 coming in and start synthesizing or constructing those complementary strands. It's going to do it differently in the leading strand as opposed to the lagging strand. In the leading strand, it's going to replicate towards where the DNA has separated. Where the DNA has separated, we call this the replication fork. On the lagging strand, it's going to elongate away from that replicating fork. And that is because, again, because of another one of the requisites of the polymerase is that you always have to grow in a 5' to 3' fashion. And so when that ends, or when the molecule opens up on the lagging strand, that direction is towards the replicating fork, or the replication fork. On the lagging strand, it's away from that. And so in order to sort of help you visualize that a little bit more, we're going to go through a diagram, and then we're going to go through, there's a link to a video that just kind of brings all the process together for you. Okay, so once you've been doing that for a while, obviously you can't have little chunks of RNA, the primer, embedded in your growing strand. And so DNA polymerase has to come along, remove the primer, remove that RNA, and replace it with DNA. Because it has both this polymerase and exonuclease activity, it can go through it. It can come and it can accomplish that. Then you sort of finally will have little gaps between the different Okazaki fragments, and DNA ligase comes along and sort of joins those gaps on the lagging strand. So here to help you sort of visually, we have our DNA molecule. Helicase comes in and separates the two strands. You have the binding protein, the SSB proteins coming in to prevent the strands from rejoining, and then you have the RNA primase coming in and forming a short stretch of RNA that will then fulfill that requirement of having that 3' hydroxyl. Here, as you can see, the sort of green colored stuff is the primer, and the 3' end of the primer on the upper strand, as you can see, kind of points towards the replication fork. On the lagging strand, which is that lower strand, it points away from it. And so once the DNA polymerase comes in, the DNA polymerase 3 comes in, we'll see which direction it's going to continue adding on to that strand of DNA. Here, the DNA polymerase 3 comes in, it's sort of that purple jelly bean looking thing, and adds DNA nucleotides to the RNA primer, all while proofreading it as it goes along. So now, as you can see, the direction of the addition of those nucleotides is towards the replication fork on the top and away from the replication fork on the bottom, and that's because it has to go in that 5' to 3' direction, or it has to read the template in a 3', if you look at the top, to a 5' direction. At the bottom, you see, on the bottom strand, you see that the 3' to 5' direction is away from that replication fork. So as the DNA strand continues to be unzipped, as it unzips, it opens up new areas to be replicated, right? And so with the leading strand, as it becomes more unzipped, the DNA polymerase 3 can just continue to add nucleotides, right? But for the Okazaki fragment, since it's going away from the replication fork, it'll have to open up a little area. The RNA primase has to come in, form the primer, right? And then the DNA polymerase 3 has to come in and add nucleotides, but it can only go a certain distance to a point where it hits up against the back end of the Okazaki fragment that had formed previously. Now it's time for DNA polymerase 1 to come in and remove the RNA nucleotides or replace them with DNA nucleotides. And then also for ligase to come in and fill the gaps between the Okazaki fragments. Here's a slide that kind of shows you everything that's going on kind of all at once. Up in the upper right, it's important to remember that there are actually two replication forks because it's bidirectional, and on the replication fork there on the left you have the leading strand on top and the lagging strand on bottom, but going the other direction because of the polarity of the DNA molecule, you would then have the lagging strand on the top and the leading strand on the bottom. Also, here is a link to a video on YouTube, a minute or so long, that just kind of brings the whole process. It's an animation, but it brings the whole process into sort of a continual thing, and it's a really video to help you just kind of understand what's happening. So that's DNA replication. Here are a few of the key points to remember. A few more. Okay, and finally, here's another link to a kind of cheesy video that you can watch to help you study DNA replication, maybe right before you go to bed or really late at night. Okay, that's it.